So hello everybody, welcome to Broad 101. My name is Megan Rokoff and I'm one of the members of the Communications Department. And the Communications Department has been organizing these Broad 101 lectures for a reason that we're really excited about, which is that one of the things we really like about the Broad is how all of our employees are trained in these really different specialties. And we wanted to create a mechanism by which we could present the research that's going on here at the Broad in a way that is accessible to everyone, regardless of their specialty. And because we'd like to make this accessible to everyone, one of the things that we wanted to ask you guys to do is to tell us about which days and times for these lectures would be best for you so that we can make them accessible to the most number of people. So we have a little survey sitting on the table, so on your way out, before you grab breakfast, if you could fill out that survey and give us that information, that would be really great. So without further ado, I'm going to introduce today's speaker, Ann Carpenter. Ann is the director of the imaging platform, and she'll be telling us today about how the platform is using software that Ann designed to extract very large amounts of information from biological images.
Thanks, Megan. Does it sound like you can hear me okay? Great. I'm pleased to be here today to tell you about the work that's been going on in the imaging platform. We have a great team of people working together to extract information from biological images. And I have to say that I just got back from giving a lot of talks at universities and at conferences, and I spent probably 5 times as much time preparing for this talk to make it accessible and interesting to non-scientists. So I'm glad that there's a good crowd here and another group of people at 320 Charles to hear about the work that's going on in our group. So just for orientation to see where we fit, our group fits within the Broad structure. As you may already know, the Broad consists of a large number of faculty, that 6 of whom are in this building, but most of whom are throughout the Boston area. We work together with those faculty. They gather here usually weekly for meetings in the Broad programs, and these cover areas like cancer, metabolism, chemical biology, infectious disease. And lots of great ideas get spawned between these faculty members coming together along with these technical expertise that are residing within the Broad platforms. And that's imaging is one of the platforms here. We focus on developing new technologies that can be applied to large-scale biology projects that are a result of all the brainstorming that goes on with these faculty members within these programs. So one of the ideas I'll focus on today comes from the infectious disease program. So Deb Hung is a core faculty member here at the Broad, and she and her postdoc Sarah Stanley had an idea for a new way to find drugs to treat tuberculosis. It's a huge health problem. A lot of people have latent infection of tuberculosis, and it's a big killer worldwide. So what was their idea? To understand what their new idea was, you have to understand a little bit about the old-fashioned way of discovering antibiotics. And so I'll show that here first. The old-fashioned approach to finding antibiotics is to put bacteria into individual wells of a multi-well plate. So I'm showing the bacteria here as green. It's basically a little test tube, and this is a multi-well plate that has a large number of little test tubes in it. So within each well, you add a different chemical. So we have a library of 1 million chemicals. Each chemical goes into a different well, and then you very simply look for the wells where the bacteria are not growing very well anymore. So now you found a drug that kills bacteria. One problem is that you've possibly found a drug that maybe also kills human cells, and that's not ideal. And as well, this is great for killing bacteria in a test tube, but that's not exactly the location where you're most interested in killing bacteria. So so there's a different approach you could take that would be actually much more effective, but probably not ideal. And that is to discover antibiotics in a more physiological system. So you take a lot of nice little girls, you add 1 million test chemicals, each chemical in a different one, and then hopefully you can figure out a way to measure how much bacteria is in each girl, and you can find one, oh, that whatever chemical we gave her is actually a pretty good one. Or if that's too complicated to measure, you can just wait a while and find the answer to your question. Okay, so that's effective, but just not ideal. So they came up with an alternate approach, which is, has the advantage of being not evil, which as scientists, we're a little sensitive to the evil scientist thing. So, so their idea was this. What if we could put human cells, these are the blue ones in this picture, what if we could put those in a dish, we could add the bacteria, they're glowing green here, and we could look for drugs that actually reduce the growth of the bacteria. And this would have a few advantages. First of all, okay, it's not whole human beings, but at least it's human cells. At least it's a little more relevant. As well, we can look, if we do this kind of a test, we can find drugs that first of all, kill the bacteria, but we can also tell at the same time, are they killing the human cells? Because if the human cells are all decimated, that's probably not the ideal drug either. But the third thing we can tell is we can find drugs that actually maybe don't do anything to the bacteria, but actually increase the immune system of the cells, help the cells to ward off the infection, if that makes sense. So these guys don't have an immune system, they're just isolated cells, but the basic idea is there that you're trying to let the cells fight off the infection on their own. So we could find all kinds of good drugs this way. And so this is what this would look like. We put bacteria and human cells in individual wells and multi-well plates. And we use special plates where the bottom of the well is clear and visible, so you can hook a microscope up to it, and you can snap a picture of what the cells and the bacteria look like through the bottom of each well. So again, we add the test chemicals, one chemical in each well. We have robotic microscopes, and this kind of work is done in the chemical biology platform here. They have all the automation to prepare all these samples, because you can imagine we're not doing this by hand anymore with 1 million samples to test. We have these robot microscopes, and what we end up with is usually more than 100,000 images in a particular experiment. Each image has somewhere around 1,000 cells, as well as all the little bacteria growing in it, and our goal is to look for the one drug that causes the infection to not take place. So that's fantastic. Like I said, all this is automated with chemical biology's really nice robotic systems. The problem is this last step, when this project came to our attention, this last step was not automated. So the only solution was to have a lot of Sarah Stanley looking at all these images by eye. And okay, maybe it's not so hard. It wouldn't take so long if the difference were this obvious, because you could scan through quite a lot of images by eye. The problem is that, first of all, it's tedious. Secondly, it's slow. But most importantly, it's not very quantitative. So you can tell a very big difference between bacteria and no bacteria, but actually in the real experiment, you see much more subtle changes. And we want to detect drugs that have even a slight effect on this process, because then we can improve them over time and come up with better drugs. So we have to automate that last step so that we don't have to drive Sarah crazy looking at images for 6 months. So how can we do that? We use automated image analysis, and that's the area of expertise of the people on my team. And so what we do is we take this original image. What we need to do is we need to first of all find the human cells, and then we need to find the clumps of bacteria, and then we need to count the number of bacteria that are within each cell. So how can we accomplish this? Well, first you have to understand a little bit about what an image is. So this is an image coming from the microscope, just shown in a black and white version. And what's actually behind that image are a lot of numbers. And so these are the digital images that come from the microscope are very similar to digital images that come from your camera, except usually we take them in a black and white form. And it's a little simpler to understand as well, because you can see the part that I selected here, the background has mostly zeros, and the part of the image that's bright has higher numbers. So this is a really important concept to understand the rest of the talk, is basically that the background of the image has very low numbers, and the cells of interest have higher numbers. So this guy has very high numbers. If you look at this one, it's a bit more dim, so it would have numbers a little bit intermediate. Okay, so in order to, so you can imagine a very simple solution to find the cells in an image would be, well, let's just, the computer is very happy to look at numbers all day long, and it does it very quickly. So maybe we can just tell the computer, here in this particular example, the cell is mostly 255s, and the background's mostly zeros, and there's some intermediate values. Maybe we can just tell the computer to, any number over 50 must be part of a cell, and any number that's under 50 must be part of the background. That would be a very simple solution. The problem is it doesn't work very well on real microscope images. So this is this is an exaggerated example, but it shows you what a microscope image might look like. So let's say, for example, that the background here is maybe zeros and ones, whereas the cells are maybe 20 or 30 are their values. So that method I just described would work well if you pick maybe a threshold of 10, and then you'll find the cells just fine. The problem is you go to the center part of the image, and the background's much brighter. Here the background is maybe 30, and now the cells are maybe 60. So picking that threshold of 10 doesn't help you in the middle. Suddenly you've got this entire gigantic blob is identified as a whole cell, and that's not going to work very well. So how do we manage to get around this? Well, what we do is we pass a filter across the image, and this is a computational thing that happens. This is sort of just a schematic to show you what it's like. We pass the filter over the image, and what happens is We measure within, wherever that little filter is landing at the moment, we measure what's the minimum pixel intensity, what's the minimum number that's within that region. So if that little square box is up here, the minimum regions are 0, maybe 0 or 1. But as it gets towards the center, that minimal region is 20 or 30. And so when that filter is passed over, you end up with, this is the result of that filter. You end up seeing the pattern that's here. And why is that useful? Because you basically just mathematically subtract the numbers here from the numbers here, and you end up with a really nice flat image. So that's probably the most complicated thing I'll describe. If that made no sense to you, stick with it, because I'll just show you what it does. So it doesn't matter what the data is that I'm looking at here, but what I really wanted to show you is that each one of these little tiles is an individual image from the microscope. And you can see that there's, especially when the red box isn't there, you can see that there's this pattern. It's brighter on this side and darker on that side, like I was showing in the other image. And so that really needs to be corrected in order for the numerical measurements, the intensity measurements that come out of the microscope, out of the image analysis to be accurate. So what we do is we correct this data so it looks very flat. And as you can see, the data that used to be a little bit mushy here is now much more clean. So the data quality is much better. All right, so now we have a very nice flat image, and the background's very black. Let's say now it's all zeros and ones or so. If we want to think about how the computer should identify where are the cells in this image so that we can count them, you can depict the image like this, where it becomes little mountains wherever the numerical values are high. So in other words, wherever it's bright. So this is probably the brightest cell, and this one maybe as well. So these two are very high peaks, whereas this guy is very large and broad, and he's a little bit more dim. And you can see as well, there's a bright cell here and kind of a dimmer one right next to it. That's this guy here, and the dimmer one is sort of hidden behind him. So one way to accomplish this now is to just imagine you're just slicing through the image and you're just taking a flat horizontal plane across it. If we take that horizontal plane across it, we could identify cells basically like this. Does that make sense? We chop off across the three-dimensional structure, and this is what it would sort of look like if we looked at what's left behind. So this is not so great. We're not really counting the cells. We have 1, 2, 3, 4, but this is 5 and 6, when actually those should be divided up into 2 individual cells, right? So this is not ideal, but the problem is if I try to take a slice lower, if I just go back to here and slice a bit lower, I end up getting this entire thing identified as a cell, so that we're not really getting the edges very accurately, and we're getting these two guys lumped together. If I want to separate those two, I would have to slice higher, but then I would miss this guy altogether if I take a slice higher. So there's really no place that you can just slice the image and have the measurements come out accurately, or have the identification come out accurately. So how do we solve this problem? There is an algorithm that was developed and worked on by Carolina Walby. She's not in our group, but she was working overseas on this problem for her PhD thesis. And one of the things that she and a lot of others in the field have been working on is to identify the maximum points in the image. So if we take this image and instead of just trying to slice it and getting all the cells stuck together or missed altogether, what if we instead just tell the computer to look for points within the image that are the highest points? That's mathematically fairly straightforward to do. And so what that does is you get 1 point at the top of each of these little mountains, including this little hidden one that's very dim behind this brighter one. And then we run what's called a watershed algorithm. And what that means, you can envision it like basically shooting water up out of the top of each of these little volcanoes. And the water rushes down all these little hills. And where the water hits between 2 independent volcanoes, that's where you're going to draw a line. And how well does that work? Well, it turns out it works really well. So you can see now we have a much better, and also if you hit the background, you should stop. The water should stop, right? So we get a much better outlining of the whole cell, and we also are able to split the 2 guys that are near each other. So this is, that's fundamentally it. That's the algorithm that we're using to identify individual cells in the images. So when we got this project into the group, Martha Vokes worked on it. She's an image assay developer in the group, and she used the software that's open source that we've developed called Cell Profiler. And she used that algorithm I just described to find cells. This actually looks pretty straightforward. There's maybe only one that was a little clumpy to begin with, that we had to use the special trick of finding the peaks. But this all happens, once she sets it up, it all happens very automatically. So she'll test it on maybe 10 images and then run it on 100,000 images without any intervention. What we do is, I was cheating a little bit. These are actually not cells, they're actually nuclei. The nucleus is the center of each cell. And we don't have the cells labeled in the image. We don't have any fluorescent dye to show you where the edges of the cells are, but it doesn't really matter, because all we really want to know is, are the bacteria, how many bacteria are nearby each nucleus. So we make these sort of fake outlines of the cells where the cells really ought to be. And then separately, we use similar methods to find the bacteria and divide up the little clumps of bacteria. And we overlay the lines that we found here, we overlay on there. And then it's a pretty simple matter to count up the number of bacteria. And when I say count, we actually, we do record how many clumps. So this particular nucleus right here has 2 little clumps growing within it. But we actually also count the intensity of the bacteria. So maybe two tiny clumps that are really dim shouldn't count as much as 2 gigantic clumps that are really bright. So we count up those numerical intensity values to get a measure of how many bacteria are growing in that sample. So I wanted to show a little bit about how the software looks that we use. And this project originally started with Ray Jones. He was a PhD student at the Computer Science Artificial Intelligence Lab right across the street in the crazy Stata Center building. He and I worked together to get this software package up and running and release it to the public. And basically, how it looks is that if you go to the website, you download some examples, you'll get a pipeline of different image processing steps. And each of these steps here are some of what I described before. You would identify the nuclei, you would identify the bacteria, you would measure different properties of the cells and of the bacteria. And you would cycle through all 100,000 images. And we actually usually use the cluster at the Broad to do that processing because it takes about a minute or so for each image that we process, and if you have 100,000, that's a lot of minutes. So we send it off to the cluster of computers, and each computer processes a different image, and it goes much, much quicker that way. So you can download the software for free from the web, and it's actually simple enough that you can load it up and run any of these examples that we have posted there. And so I'll show you a couple of the examples that are in completely different fields. We focused on Sarah's project so far, but I'll show you a couple other of projects we've done in the past. This is a project that I worked on when I was a postdoc at the Whitehead Institute right next door. I worked with Leah Cohen, who is in Sue Lindquist's lab. Sue Lindquist has been doing her sabbatical here at the Broad over the past year, which is the 2 institutes closely interact with each other. In their particular project, they were interested in how yeast grow, and they were looking at particular mutations and particular drug-resistant yeast, and they needed to count up how many tiny colonies and how many medium-sized colonies and how many large colonies. And the problem is these are those plastic plates, plastic agar plates. Each plate has probably 1,000 little colonies on it, and using your judgment to determine what's small, medium, and large gets really annoying over time. And so what we did is we set up the software, and this is one of the examples you can download, that finds each individual colony and then classifies them according to size. So the yellow are the largest, and red are the medium, and the blue are the smallest ones. In another yeast-related project, we had these square plates that, again, had agar growing in them and little patches of yeast, where each patch had a different chemical growing in it. And you can, if you wanted to do this by eye, you could certainly do that, and you could say, okay, it looks like these yeast aren't growing as well in that particular chemical, so maybe that's a good antifungal chemical that I found there. But it would be very nice if you could just automatically find them all and get the numerical data out, quantitative data, so that you can get, at the end, you run all the images through and you get a rank-ordered list. These are the strongest chemicals that can prevent the growth of these particular yeast. I'll skip that one. So what I've shown so far are fairly straightforward things where you're just counting or you're measuring size in images and so forth. And I think hopefully by now you have an understanding of how those algorithms actually work behind the scenes. So those methods worked very well for a large number of projects that we worked on, but sometimes we would get stumped. And the reason we would get stumped is because when a biologist looks at an image of cells, there's so much information present there, and you're able to discern patterns and see things that the computer is not necessarily knowing what to look for. And so, as an example, we worked together with, on some images that had been created by Dave Root and the RNAi platform, and especially Serena Silver and Jen Grenier and Jason Moffett at the time had prepared this very large, beautiful set of samples. And the set of samples they imaged by microscopy, and what they discovered, as you may know, RNAi is a method for perturbing a gene in the genome. So this is sort of schematic here, but if you have the chromosome, you perturb a particular gene. Some genes, you perturb them, and the cells suddenly look funny. So this particular gene, for example, this is what cells normally look like. In this particular gene, the nuclei are somewhat crescent-shaped. If you knock down that gene, suddenly the cells, I have no other way to explain it other than they look like they're going somewhere. If you knock down this gene, the cells have these cute little, they grow in this cute little straight line, so they look like little peas in a pod, and all kinds of other things. So in this image that we had found, just stumbled across serendipitously, we stumbled across all these funny little phenotypes, funny little appearances or morphologies of cells. And so we thought, well, we've got 40,000 images. The experiment tested a lot of genes. We've got 40,000 images. And ideally, we would like to know every single one of the genes that were tested. We'd like to know every single one that causes this particular appearance of cell. And we'd like to know every single one that causes this particular cell type. And to look through 40,000 images for 15 different cell types was going to be a really obnoxious project, and nobody wanted to do it. So that's where Ray Jones thought of some of the machine learning that had been popular in other fields, non-biological imaging fields, and he thought, let's see if we can apply this to biological image processing. And this is where it gets very cool. This is where the computer is actually learning from the biologist what to look for in the images for these very complex appearances that the computer otherwise can't discern. So as you remember, we've got thousands of wells in the experiment. So we have tens of thousands of images. Each image has maybe 1,000 or so cells growing in it, which means we've got 10 million cells per experiment. It's certainly far too much for us to score by eye. And as you now know, we can use Cell Profiler to find each cell and to measure everything we can think of to measure about it. And so what we end up measuring is actually usually more than 500 features for every individual cell. So what are these features? They're things like, what's the shape of the nucleus, the blue part of the cell? What's the shape of the red part of the cell? What's the size of those two different parts? And size would include things like area and perimeter and the length and the width. We can measure the texture of the staining. How smooth is the red staining? How patterned is the blue staining, and so on. So we make all these measurements, and I'm going to depict those measurements here just schematically as sort of a barcode, that this particular cell has this particular set of features. It's actually 500 rather than just 30 or so here, but you get the idea. These are the measurements that we've made from this particular cell. And I especially want to thank Bang Wang and Nadav Kupik and Chris Lewis, who put together these illustrations, which will go into a publication fairly soon. So how does it work? So once we've made those measurements, that's the part you know about already. So how does the machine learn how to look for what the biologist is looking for? Well, the software presents a few hundred cells to the biologist to score. And so she looks at each of those images, and she decides whether that's interesting or not interesting. So if she's looking for crescent nuclei, she'll sort crescents yes and otherwise no. In this case, it looks like she's looking for projections, but they have to be blobby projections, not pointy projections. So she's sorting. And meanwhile, she is looking at the images. She doesn't care one bit about the numerical measurements that come along with each cell. But the computer doesn't really care about the image. The computer cares about these measurements that have been made, because the measurements are what tells the computer what to look for. So over time, as the biologist sorts these two different classes of cells, the computer starts to figure out, hey, it looks like all the cells that you are counting as positive, they're maybe a little high, they have a somewhat higher value of this particular feature that we've measured. And the cells that are negative have maybe a little lower value of this particular feature that we've measured. So the computer is looking at all these barcodes and discerning what's unique about the positives compared to the negatives. And as soon as the computer has a reasonably good idea of what the biologist is looking for, the computer then sends some images back to the biologist and says, okay, I think these 20 images are what you're looking for. And so the computer then sends back more images for the biologist to look at, and the biologist says, okay, there's 20 images here that you think are positive, but only 15 of them are really positive. These other 5 are negative. And so she'll sort some more, and the computer will learn from that. And over time, with this iterative cycle of training, the computer gets a really good sense of what it is that the biologist is looking for. And so once the computer knows what it's doing, we go to the automated scoring, where the computer has its rule, which by now is a really good rule for defining the cells that are positive, and then it scores all 10 million cells. And this takes less than 10 minutes. It doesn't take very long to do. And basically, the computer just runs each one past its rule. It compares the barcode of that particular cell versus this rule and decides whether, yeah, it's a positive, or no, it's a negative. So what this lets us do is basically for every sample, we can calculate what's the percentage of cells that are interesting, that are positive. And this is what it actually looks like when you are using the software. It's quite user-friendly, so you don't have to know anything about what's going on behind the scenes. All you have to do is say, here's some cells from the experiment, sort them in a way that you think is showing what you're looking for. So we might put peas in a pod over here and everything else over here, for example. In this case, I'm sorting cells that have a very straight green bar of the DNA in them, compared to all the others that don't. And then I do the training, and then the computer says, OK, I think I've learned it. Here's some more that I think are positive. What do you think? And so it spits those out up here, and I say, yeah, I think actually most of those are pretty good. If I disagree, I might sort some more and continue onwards. And that's how we score these more complicated phenotypes. So we've been able to do that for these 15 different phenotypes and 40,000 different images. We've been able to really rapidly determine which genes cause these different changes in the ways the cells appear. We've also used the method in a couple of other projects throughout the Broad. So you might have heard of Eric Lander. He actually works here. And his postdoc is Piyush Gupta. And he used this software on a project related to breast cancer. And you can ask him if you have questions about that project. We worked together at Harvard Medical School with Tim Michelsen's group to distinguish different stages of abnormal mitoses, abnormal cell divisions. And as well with Ricky Eggert, who's also at Harvard, normal cells have 1 nucleus per cell, but if their cell division is messed up, they end up with having 2 nuclei per cell. And they were also able to use this machine learning method to accomplish that project. So where are we headed next? Well, we have a couple of things that we're interested in working on. One is time-lapse movies. Movies are actually just a pile of images. So here's the original movie, and here's the cell profiler identification of the individual nuclei of cells over time. You can see they're dividing. And once we have identified the cells, we make measurements of them. So we can measure things like, let's look at the area. Right now it's high, and now it's going very, very low. And you'll see right now the shape is up here. It's very round. The cells are very round during this phase, but they're about to become non-round as the little red dot moves down. See, they're now getting very crinkly at the edges. And they're flashing in and out because as the cells hit the edge of the field of view, we don't want to count their measurements. So they sort of flicker when they're at the edges. And then lastly, another direction we're moving in is to look beyond individual cells, but to look at other types of biological samples. And so one of my favorite examples are using C. elegans worms. So this is a picture of a well of those multi-well plates. The worms are quite large. And our goal is to identify each individual worm because you can actually tell the difference between dead worms and live worms just based on their shape. And so can you guess in this image, there's pretty much one dead worm in this image. Can you guess which one it is? It's the one that's got rigor mortis here. It's the one that's very, very straight. So live worms are very curvy and squiggly, and the dead ones are straight as a rod. And so if we can get this working, it's still in progress, but if we can get it working, then we can tell the difference between those two, and we can score hundreds of thousands of samples to find chemicals that are affecting different processes in behavior. So worms actually have a lot more behavior than cells in dishes have. And so we can see, are they overeating? Are they starving themselves accidentally? We can look at all kinds of different features of live worms that we can't do with cells. We're also very excited working together now that the Stanley Center is here and we have a psychiatric disease program. We're excited to get analysis methods that work very well for neurons. And they look very different from usual cells because they've got such long extensions, and we want to measure how much do the neurons branch and so on. We also have projects where people are doing experiments with zebrafish embryos that are growing in dishes. And as well, we've been working a little bit on some projects where we're looking at movies of yeast, which I guess the movie's not working, but where the yeast are dividing over time, and we're measuring the properties of how the cells respond to cell division. So lastly, I wanted to just show the team that works together on these projects. You already saw Martha working on Sarah Stanley's project. David Logan has also been in the group for the past half a year or so. We have a new person, Mark Bray, coming next in a couple months. Ray Jones started the Cell Profiler project, and Vebjørn Losa is working in the group on some of the neuron projects, and is also working on some of the interesting systems biology projects that I won't talk about today. And as well, we've got Bjorn starts on Monday, and August is coming later this year. So we've got a really good team, especially I want to acknowledge the help of our two administrators, Peggy Anthony, who takes care of a lot of the infrastructure, the IT work in our group, and Christopher Lewis, who is actually a rock star, but when he's not on tour, he works together in our group handling a lot of the websites and IT-related stuff. And with that, I thank you very much for coming and for listening. And again, you can get the software for yourself and play around with it if you like. It's pretty fun to play with. And I just want to especially thank the collaborators who work together with us and give us all the images to play with, and also the funding that at the moment is coming from the Broad Institute as we get ourselves up and running. Thank you. I'm very happy to take questions.
On the machine learning algorithm, does that basically give you a yes/no answer, or can it be more quantitative than that?
Okay, so the question is whether the machine learning algorithm gives a yes/no answer, or whether it can be more quantitative. And the answer is that it actually gives you a number that can range anywhere from negative to positive. And if it's a positive number, then we count it as yes. And if it's negative, it counts as no. But you definitely can see how far away from zero is it. So is it a very strong yes or a very strong no? And so in this case, it works out best for the experiments we're doing to count them as strictly yes or no. But in other contexts, it might be useful to say, to score more how strong is it and how weak is the appearance of the cell. That's a good question.
Have you considered any extensions to three-dimensional, like say small tumors that you could look into from different angles?
Okay, yeah, so the question is, have we considered doing any work with three-dimensional images, for example, tumors and so forth? We haven't gotten into it yet, but there's a lot of the same algorithms that I described that work in 2 dimensions can actually work on a 3-dimensional structure too, so that you can get all the boundaries of a cell and measure them appropriately. And so that's something that's on the horizon. We haven't started working on it yet. We haven't had projects, since we mostly work on these large-scale screens, most people don't do a very large stack of images for every single sample, because it takes forever on the microscope. But that might be something that comes up in the future. Good question.
How much power are you having from your training data?
How much?
The power of analysis for your training data?
I'm sorry, how much?
Power of analysis? How much power are you having?
How much power are we having from
the training data that you're doing?
The training data. I'm not sure I know what you mean by power.
Like how much false positives and false negatives?
Oh, okay, what's the accuracy of it? We're actually in the process of testing that right now. Usually it's something, we've used it as a tool where the biologist does the sorting, and when the biologist is comfortable with the false positive, false negative rate, that's when the biologist stops and just scores the whole screen. So far it's been sort of ad hoc. We do cross-validation, and so every biologist has a different balance of what they want in terms of, some people come in and say, I don't want to miss anything. I don't mind if I have to look through 1,000 images and maybe only 100 of them are real. I just want to make sure I don't miss anything. Whereas others say, okay, I don't want to look through 1,000 images. I don't want to have a lot of false positives, but I don't mind missing a few as long as I get the most of them and most of them are accurate. So you can basically decide for yourself what you want the balance to be.
So do you find that the staining adds a lot of variability, or is that one of the things that the biologist kind of has to normalize?
Okay, so the question is whether the staining adds a lot of variability to the measurements. And the answer is yes, and it's incredibly boring to try to fix that kind of problem, so I didn't talk about it at all. But yeah, when you've got these robots, in theory, the robots are very uniform and they're working very repetitively, and so they should be as close as possible to each other. But the reality is that the first 5 plates might be stained a little brighter than the second 5 plates, and so that's something that you have to run some statistical techniques to make sure that everything's exactly uniform. Yep. Great, thanks everybody for coming.